All right, it's my pleasure to introduce Sean. He's a second-year master's student in the Aquatic Animal Health Laboratory at Michigan State University and is advised by Dr. Tom Locke. He is conducting research aimed at preventing the vertical transmission of Flavobacterium psychrophilum, the causative agent of bacterial cold water disease in trout and salmon, through serotypic analysis, rapid diagnostic assay development, and the evaluation of novel egg disinfection protocols. So take it away, Sean.
All right, thank you so much for that. Before I get started, I want to go ahead and say thank you to all of my co-authors for their contributions. And today I'm going to be talking about Flavobacterium psychrophilum, which is this yellow pigmented gram-negative bacterium shown here. And I'm very excited to share some of my current research findings about the serotypic variation, or essentially how a fish immune system views this pathogen in the US, which is something that it began to be looked at decades ago, but since then, it really hasn't been examined on this scale or using the tools that we now have available today. And then in addition, I'll be talking about a new diagnostic assay that we're developing in order to screen for this pathogen in the field. So before I get into my work, I want to highlight the importance of aquaculture, both globally and in the US. Aquaculture is the fastest growing animal agriculture industry globally. And so traditionally, we've obtained fish for human consumption through capture fisheries, which is shown here in the gray line. And we can see in this figure that aquaculture in the blue line has grown tremendously since the 1980s, to the point that around 2015, aquaculture actually overtook capture fisheries. And it's now the primary way in which we obtain fish for human consumption. And so this industry is also growing rapidly in the United States, where trout are especially lucrative. And here we see that in 2019, we tried to raise 79.2 million trout, but we only actually ended up selling 47.2 million of these. And this is because disease can be a major impediment to the productivity of this industry. So in 2019, we lost over 1/3 of all trout produced to disease. And of the pathogens that are responsible for these losses, pathogens within the family Flavobacteriaceae are especially problematic. And then of these, Flavobacterium psychrophilum is one of the most damaging. So this bacterium, like I said earlier, is the causative agent of bacterial cold water disease. And as you can see in the image, this yellow pigmentation is from these mats of bacteria all over the fish. And in severe cases, it can actually cause the flesh of a fish to erode away to the point that it can expose the vertebral column, as you're seeing here. And if this disease goes unchecked, it can result in losses of greater than 70%. So why is it that bacterial cold water disease is so problematic? There are a few different factors that are playing into it. One is that we're seeing mounting antibiotic resistance against the only 2 antibiotics that are currently approved for use against psychrophilum in the US. Another is that there's no commercially available vaccine against psychrophilum approved for use in the US. And this pathogen is able to be transmitted both horizontally from one fish to another, as well as vertically from parents to offspring. So this means that the eggs can already be colonized by psychrophilum, and then this can result in disease in the fry that hatch. And so some of these problems may be influenced by the intraspecific diversity of psychrophilum, both genetic and serotypic. And we already know quite a bit about the genetic diversity of psychrophilum. Our lab, as well as other researchers, have done a lot of work on this. And so some of you may have seen this figure before, and it's definitely complex, but thankfully, the only thing that I want you to take away from it is that every single number here represents a genetically distinct strain of psychrophilum as determined by multi-locus sequence typing. And so this can have implications on the geographic distribution, host specificity, antibiotic resistance, and more. But unfortunately, genetic diversity is not necessarily representative of a fish immune response. And so here's my representation of psychrophilum here. And my work focuses more on serovariation, which is the result of differences in psychrophilum surface proteins, which are shown by these different colors. And this can actually affect how a fish immune system recognizes the pathogen. So before I get into what we know about the serotypic diversity of psychrophilum, I want to highlight why serotypic diversity can be so important for vaccine development, using an analogy with the current SARS-CoV-2 pandemic. So we've heard a ton of talk about the vaccines that have been developed against COVID-19, as well as the different variants that keep popping up. And the current vaccine was developed to combat the original version of the SARS-CoV-2 virus. And we know that it works quite well against it. But now the big question is whether or not this vaccine protects us equally as well against the other variants like Delta, Omicron, and Epsilon. And it's actually a similar concern for potential Flavobacterium psychrophilum vaccines, which is just one of the reasons why understanding the serotypic diversity is so important for us. And so there was a good amount of pioneering psychrophilum serotyping work done in the US in the 1960s through the 1980s. But then in the 90s up through now, most of this work is shifted over to Asia and Europe. And the concern with this is that based on genetic analyses, psychrophilum strains are very distinct between the US and other regions. In addition, these previous methods of serotyping psychrophilum, which primarily were through ELISA or slide agglutination, they were quite complex, they were expensive, really time consuming, and they required specialized reagents. And so because of this, there was a need for a standardized, straightforward assay for serotyping. So thankfully in 2017, there was a multiplex PCR-based serotyping scheme devised, and then this was later improved upon in 2020. And so this table is from the Roche paper, the first one that I just showed you there. And for those of you that are familiar with psychrophilum serotyping, you may recognize these FP and TH types. These have really kind of become the standard that we see when referring to psychrophilum serotyping. And in general, we've seen that the number of serotypes ranges from about 4 to 8 types, depending on what method you use. And so Roche wanted to create a standardized method that was easily comparable between labs, and they were actually successful in doing so. So this table describes how the previously identified serotypes through ELISA and slide agglutination correspond to which of the newly identified molecular serotypes shown here. So in essence, they were able to find genetic identifiers specific to the previously identified serotypes and then create PCR primers capable of identifying them. So the building off of this, the 2020 Avendaño Herrera paper that I just showed you was then able to find similarities between some of the isolates within type 0. And out of that, they identified type 4, which brings us to the 5 currently recognized molecular serotypes. And it's through this multiplex PCR method that I've done my work. And so that brings us to the overarching objective 1, which in light of the lack of a vaccine and the mounting antibiotic resistance, the overarching goal has been to elucidate the serovariation of Flavobacterium psychrophilum in the US. And eventually this will lead to, this will hopefully be able to inform vaccine development in future studies. And so using this multiplex PCR scheme, so far I've serotyped 324 US isolates, and these have originated from 17 different US states, and they've been collected over the past greater than 40 years, so it's been a huge project. And the way that this works in brief is that I first had to revive these isolates from a cryogenic stock, and then I would perform nucleic acid extractions on them and quantify the extracts. And then for every single isolate, I ran this multiplex PCR assay. And then finally, I would analyze these isolates through gel electrophoresis. So every time that I did this, I had my 6 controls, which were composed of the 4 distinct molecular serotypes, which are types 1, 2, 3, and 4. And then I had type 0, which is the catch-all group for isolates that don't fall into any of the other distinct molecular serotypes, as well as a negative control. And then I had my unknowns, and based on the band size of the controls, I was able to match the unknowns to a control, and then I could identify the molecular serotype that way, which remember is based on the previous serological characterization. So for example, this unknown in the blue box would be a type 1, and then the unknown in the purple box would be a type 2. And so I got very, very good at identifying these different serotypes after running, I don't even know how many different gels. And this image doesn't even show all of the gels that I've ran. So before we get into my actual findings, I wanted to provide a little bit of context for what our prior knowledge for molecular serotype was like in the US for psychrophilum. And before my work, we had multiplex PCR information for 14 US isolates. And based on that, this pie chart represents the scope of our understanding of the molecular serotype diversity of psychrophilum in the US prior to what I've done. And it's worth noting that 12 of these 14 isolates, the gray up there, fell into type 0, which as we just talked about is an unknown catch-all group. And then this represents what we now know after my molecular serotyping work. So you can see that things look quite a bit different from our original 14 isolate understanding to our current 324 isolate understanding. And within my data set, type 0 was the most common, but as I just said, we don't know if within this group they're all similar or if they represent countless other molecular serotypes. So we'll look at some of this closer, but there were some apparent host associations as well with certain molecular serotypes. All right, so over the next few slides, you'll see more figures like this one. On the x-axis, we have the percentage of isolates belonging to each molecular serotype. And then on the y-axis, we have species. It will be changing from species though, so just keep an eye on the y-axis. And then I also have the number of isolates listed on the right-hand side. And so previous findings on other continents have suggested that there may be some host associations. So I wanted to look at this for the United States as well. And we do actually see some interesting trends. So in rainbow trout, which is the largest data set that we have for any one species, you can see that we collected psychrophilum of every known molecular serotype from this species. And this is in contrast to a species like coho salmon, where we see that 90% of the isolates that we've collected from coho were type 0. But again, type 0 is that catch-all group, and it really could be any other number of unknown molecular serotypes within that. And while some of these data sets are not the largest, this still represents the first time that molecular serotype data for some of these species has been collected in the US. And for lake whitefish, lake trout, and brook trout, this is the first time that molecular serotype data has been collected for these species in general. Okay, so we also saw some interesting variation by state. For example, thus far, all 3 of our isolates from Minnesota are type 3. And this is interesting that out of all of our isolates, only 4 were type 3, and 3 of those 4 came from Minnesota. And then in California, we see fairly little variation, where over half are type 1s, while about a third are type 2. And then there's also a very low prevalence of type 0s. And then Michigan is especially interesting, as this is where the majority of our isolates have come from. And we see every single molecular serotype represented here. And Flavobacterium psychrophilum and its close relatives, they cause more losses in Michigan state fish hatcheries than all other pathogens combined. So by investigating this, we're hoping to inform rapidly deployable vaccines for use in these facilities. Our isolates are continuing to come from Michigan, from a collaboration between our lab and the Michigan Department of Natural Resources. So anytime that we necropsy fish in collaboration with the DNR, if we recover psychrophilum, it will get added into our cryogenically stored stock, and then I can perform my analyses on them from there. And so if we look at the distribution of molecular serotypes within Michigan, we can see that type 0, that unknown catch-all group, is the most predominant, followed by types 4, 2, 1, and then 3. And while all molecular serotypes are represented in Michigan, the low prevalence of type 3 indicates that type 3 might not be as much of a priority for vaccine development. And then this image shows the molecular serotype distribution for Michigan by species. So one really interesting point is that in the genus Salmo, which includes brown trout and Atlantic salmon, we see that nearly half of these isolates are type 4. So this would suggest that vaccination efforts for Salmo might need to be focused on type 4. And then we also saw that about 50% of Chinook isolates were type 1s, about 40% of rainbow trout isolates were type 2s. And this is really important to consider for the vaccine development. Also, clearly coho isolates are most commonly type 0s, but we need more research in order to determine what the variation within this catch-all group actually looks like. And then we also saw some really interesting variation by source facility in Michigan. So that's what this figure is showing. On the y-axis, we have SFH, which represents state fish hatchery, where the fish are raised to be stocked into public waters in Michigan. And then the weirs are where the gametes that are used to populate the state fish hatcheries are collected from, both wild and feral fish. And so analyses for this are still underway, but at least initially we can see that for state fish hatchery B, we may want to focus on type 4 for vaccine development, while at state fish hatchery E, we would want to focus on type 2. Now this is in contrast to state fish hatcheries C and D, which both have a lot more diversity within them. And it's also interesting that Weir 1 has a similar distribution to that of state fish hatchery F, given that Weir 1 is where the eggs that are collected to populate state fish hatchery F are from. And this shows that wild populations and vertical transmission may be able to influence the molecular serotypic diversity of psychrophilum within a facility. And so all of this means that if we know what types exist at a weir, then we may be able to be better prepared for what types may pop up at the associated hatchery. And so this work represents the first large-scale analysis of Flavobacterium psychrophilum molecular serotype in the US. But why does that actually matter? Well, we touched on it earlier, but this information will help to inform vaccine development against this devastating pathogen. And we've shown that there's substantial molecular serodiversity both in the US and in Michigan, with a heavy predominance of type 0. We also saw that some types are more commonly associated with some species over others. And while it may be possible to develop a one-size-fits-all vaccine after further research, most immediately, this information can help to devise more tailored and rapidly deployable vaccine approaches in the form of whole-killed or autogenous bacterins. And so because of this, the serotyping data could be invaluable when it comes to combating this pathogen within a given facility. And so in addition to vaccine development, this information is already in the process of being used to create a new diagnostic assay for psychrophilum that we use field side or pond side. And so that brings us to my second overarching objective, which is to use existing genetic data combined with the newly derived molecular serotype data to develop a rapid, sensitive, and specific novel diagnostic assay to screen fish reproductive fluids for all known US psychrophilum variants under field conditions. So the point of developing a diagnostic assay that can be used in the field is so that hopefully we can prevent the vertical transmission that I mentioned earlier. And so if we can quickly tell if eggs or sperm are infected with psychrophilum, then we can avoid using them and avoid vertical transmission. So an assay that's ideally suited for these purposes is loop-mediated isothermal amplification, or a LAMP assay. And this assay occurs at one temperature. It doesn't require expensive reagents. And you can use a color indicator in order to discern positives from negatives just by the naked eye. So this means that we can perform this reaction in something like a water bath or a heat block. And then in less than an hour, we can tell if psychrophilum is present by simply looking at the tubes. So in this example, we can see the positives in green while the negatives are orange. And before I get into the specifics of actually developing this assay, I want to briefly explain how LAMP works. The basic concept is that we have 4 to 6 primers based on a specific target region. And then we have one of the inner primers invading the segment of DNA, and it's then extended, thereby splitting the double-stranded DNA. We then have one of our outer primers come in, and that separates the strand of DNA formed by one of the inner primers. And this ends up forming a loop structure due to the reverse complementary sequences. And then this will end up repeating on the other side as well, which forms this characteristic LAMP dumbbell structure. And from here, exponential amplification can occur, forming a variety of different structures that can all be detected by multiple different means. And so that's the actual mechanism, but what all of this means is that LAMP is oftentimes more sensitive and more specific than PCR because we have 6 primers instead of the normal 2 for PCR. And it also happens at a much faster speed than PCR because there are so many different binding sites for these primers to work off of. And so step 1 of creating this assay was to design primers that were specific to Flavobacterium psychrophilum and were also capable of recognizing every known psychrophilum variant in the US, which at the time of design was 504 genetically distinct variants. And so there's a website called PubMLST that hosts sequence information for multi-locus sequence typing. And MLST is based on 7 housekeeping genes. So to begin, I downloaded sequence information for all of these isolates and for each of the 7 housekeeping genes. And then I aligned these sequences using the primer blast feature of the NCBI website. And I found that of the 7 housekeeping genes, only one, which was gyrase B, seemed promising. So this alignment allowed me to identify every single base pair discrepancy between each of these isolates, and those are shown in red by these arrows. And so I then recorded the position of these discrepancies, and I ended up with a list of specific base pairs to avoid when I was designing my LAMP primers. And then I put this information into the Primer Explorer version 5 software, and I was able to produce 2 potential primer sets based on the gyrase B gene that at least in silico were capable of amplifying every known US psychrophilum variant. We then ordered these primers and then began lab testing. So if you're familiar with PCR and gel electrophoresis, this gel might look like a mess, but this is actually what you want to see when you're running a gel for a LAMP assay. All of the different products that are produced from LAMP result in this characteristic banding pattern. So when I tried these primers in the lab, I found that it clearly amplified all 5 molecular serotypes, and it didn't appear to have any reactivity with the non-target close relatives of psychrophilum. So now I needed a means of differentiating positives from negatives by the naked eye, and I ended up coming across a color indicator called hydroxynaphthol blue, which would change from purple to blue as free magnesium ions are used up in the reaction. And much to my relief, this ended up working really, really well. So you can see in this image that all 5 molecular serotypes show up blue as positives, and then the non-target and negative tubes are purple. And this assay seemed to work under ideal conditions and using DNA that was extracted using a commercial kit. But the whole reason why I'm creating this assay is for it to be used in the field. And because of this, I needed an easier, cheaper, and faster way of extracting bacterial DNA. So I tried 2 very simple methods. The first one was just boiling, and the second one was actually microwaving, which I know sounds a little bit goofy, but bear with me because actually both methods appeared to work on the gel. However, boiling was the only method that actually worked in terms of inducing a color change in the tube. So because of that, boiling was selected to undergo further testing with fish reproductive fluids. So in order to test the efficacy of this assay with fish reproductive fluids, I inoculated or I spiked both fish ovarian fluid and milt or sperm with Flavobacterium psychrophilum, and then I boiled the reproductive fluids, and then I used that as template DNA for the LAMP assay. So on this image, I have a ladder in the first lane, then spiked ovarian fluid, non-spiked ovarian fluid, spiked milt, non-spiked milt, spiked water, non-spiked water, and then a positive and negative control. And then I also have a zoomed in image of the associated tube for each of these overlaid on the image. And I found that this method seemed to work really well for ovarian fluid, shown in the box here with the positive and the negative ovarian fluid. But this method didn't seem to work right away with milt. So given that millet is extremely cell dense and it's full of proteins and genetic material, this wasn't overly surprising, though it was a bit disappointing. Thankfully, though, my advisor suggested simply diluting the boiled millet extract and seeing if that helped. So I gave that a shot and it actually worked perfectly. So you can see in this image, I have spiked millet in the top row and then not spiked millet in the bottom row. And I found that by diluting the millet extract anywhere from 1 to 5, all the way up to at least 1 in 50, we were able to visually differentiate the positives as we intended. And so again, much of this work is because vertical transmission is a major problem for aquaculture facilities and hatcheries. And because of this, there's a need for an assay like this LAMP assay to allow us to test reproductive fluids in the field. There's certainly more work to be done, but so far it's amplified all psychrophilum variants that we've tried to date, and it functions for reproductive fluids in the lab under mimicked field conditions. So next we'll be estimating the limits of detection for this assay. Then we'll also see how it works in naturally infected samples. And then we'll go into the field and run this assay alongside the current standards of culture and qPCR. And so all of the hard work that others have put into understanding the genetic diversity of psychrophilum, in addition to the serotyping work that I took you through earlier, which represents the first large-scale serotyping analysis of US isolates, this will end up informing future work into vaccine development and is informing this assay, all with the goal of breaking vertical transmission. All right, so with that, thank you so much for having me, and thank you to the USDA NIFA for being the funding agency. Thank you to everybody involved in this project, and I would be happy to take any questions at this time if we still have time.
We do have some time. Excellent presentation, Sean, an incredible amount of work that you've done.
Thank you.
We've got a couple of questions in the chat, but feel free, folks, to unmute yourself as well. One of the questions, did you look at the antibiotic resistance difference between the serotypes? Is type 0 resistant to the antibiotic?
You know, I don't believe that work has been done yet. I could entirely be wrong, but as far as I'm aware, this, like I said, my serotyping work is the first large-scale analysis for US isolates. So there may actually be some information for antibiotic susceptibility by type outside of the US. For the US, though, as far as I'm aware, that isn't work that's been undertaken yet.
And along the same lines, did you look at trends over time to see if predominant serotypes changed over the decades?
I did actually, specifically for Michigan, because we have all of the hatchery and weir information. And so that's one of the things that I still need to put more work into analyzing it and gleaning out any trends that there may be. But we definitely do see like year-to-year trends. And I can't remember which type it was, but at one of the facilities, I actually saw like every other year there would be a spike of a specific type, and then it would go away the following year, and then it would jump back up next year. So yeah, there's certainly interesting stuff to be seen there.
Okay. Jim Winter.
Hi, that's a great talk. I'm wondering, were all those isolates from active disease outbreaks, or do you have any information, I guess, about the serotype in relation to pathogenicity or virulence amongst those different serotypes?
Yeah, so actually all of those isolates, for the most part, they were collected from different disease outbreaks in Michigan. Like the Michigan one specifically come from anytime that there's like a die-off in fish, the DNR will send us their fish, and then we'll look at them, and if we get psychrophilum, we'll keep it that way. In terms of seeing if any specific type is more virulent than another, we've seen some host specificity associations, but it's really like with all of my work into looking at all of these different isolates in other continents, like there's a lot of work that's been done in Japan, and they were able to see some relatively clear host associations. As soon as we got to the large scale analysis that I've done, it's a lot more difficult to say that there are host specificity when it comes to the individual serotypes. I'm not sure if I explained that very well, but basically in the US, there's a lot more variation than what we've seen elsewhere.
Right, it looked like, yeah, it was kind of a patchwork, and yeah, great information. It seems like all those isolates are potentially, or all those different serotypes are potentially important pathogens because they all came from active outbreaks.
Yeah, absolutely. And that's another side of this, is that all of this has really been circumstantial. You know, we collect isolates as we're able. So that can definitely end up having a sort of filtering effect on our end as well.
Thank you.
Of course.
Jim Winton, you have a question?
Yeah, Sean, thanks. That was really interesting and pretty important. Using your COVID analogy, do you think there's some concern that a monovalent vaccine will simply just drive the selection of the other serotypes in the population, such in areas where they're present? Or will a multivalent vaccine eventually be needed, or you just have to stay on top of this?
Yeah, it's kind of a blend of everything that you just said. So the main idea with this is that if we're seeing a specific outbreak of a specific type at a facility, and this would mainly just be for a facility while they're rearing the fish. This wouldn't be like something to vaccinate wild populations. But if we know what type it is, then we can take that directly from the facility and then pretty quickly turn around a vaccine in order to treat that specific outbreak. And so that doesn't require as many approvals through like the FDA and all of that sort of stuff, because we're collecting it from the facility.
Right.
However, you're definitely right. There could be, it may end up selecting for certain types over others. But depending on the host associations that we see within a facility, ideally that wouldn't be the case, but you're absolutely right that could happen.
Yeah, an autogenous approach, you can be more nimble, but it won't work for the corporate companies that want to produce a commercial product where they need to have testing and some stability of that product. And over time, that could just simply drive the evolution or the selection of other serotypes. So I'm wondering how that eventually might work out. Maybe your autogenous approach is actually the better way.
Yeah, well, that's the idea, is that the autogenous approach will be kind of an interim sort of thing where we can like rapidly turn it around and then treat different facilities, all while this further research is going on to hopefully have a one-size-fits-all vaccine someday.
Yeah, great, thanks.
Of course.
Great, thank you. Tom, you have a question?
Hi, Sean. Great presentation. I was I'm particularly interested in some of the preventative techniques that can be applied towards managing this pathogen. And I'm just trying to envision on like how this would work in the field at the spawning weirs. And maybe a question for Tom Locke as well is, you folks in Michigan, know what you feel for. You have like a prevalence level on how many broodstock are infected, and coming up with a technique by, potentially doing pair spawns and eliminating eggs. Can you talk a little bit on how you would envision that working? Because I think that's important. I do appreciate all the, this work on the vaccines, and hopefully we will get a successful vaccine, but it seems like that might be down the road sometime. And what can we do to do our best to try to prevent it from happening, preventing it from coming into the hatchery and affecting the hatching fry?
Yeah, so that's actually, Tom may end up having more to say on it, but that's really what the main focus of my project as a whole is preventing psychrophilum from getting into hatcheries. And so one of the ways that we're planning on doing that is through this LAMP assay that I just took you through. And the great thing about it is that, like I said, it really just needs a heat block. And so if I can optimize this boiling extraction method a bit more, then we can just bring a heat block to like a weir, and then as we're collecting fish, I can extract DNA, and then we can have the LAMP assay running alongside of their spawning. And then if we see any positives, we can throw out the positives and then just keep all of the gametes that are negative. And then that way, hopefully we'll avoid bringing psychrophilum into the facilities. But we do know that it can persist inside of the eggs. And so that's a whole other component of my project that I haven't started on yet, but we're hoping to design a new egg disinfection technique that will actually be able to remove psychrophilum from the intra-ova infection that we've seen it do.
What would you guess would be like the time frame from the time that you started that assay in the field? How quick do you think you would be able to have an answer?
I would say within an hour and a half, in terms of all things considered, like both the fidgeting of like getting everything ready and all that sort of stuff, but the actual DNA extraction, 10 minutes from what I've found by boiling. And then the assay itself, I've gotten it to work in as little as 45 minutes, but really an hour of full running is what you need. So if you're super desperate, you can probably get it done in under an hour. But all things considered, probably an hour and a half.
Thank you.
Still better than the BKD ELISA, but for the same kind of purpose.
Mhm. So, Sean, you got a couple of questions in the chat box here related to the molecular data. Dave Groman's wondering if it would be useful to develop a DNA or mRNA injectable vaccine, given the molecular data that you have now.
You know, I personally don't know. I think that is certainly something that further research could look into, and especially given that, you know, that the COVID vaccine is an mRNA vaccine. So, you know, we know that it's effective. I think that probably the biggest issue with that would be approval, because as far as I'm aware, at least in the US, mRNA vaccines are pretty difficult to approve. So that's kind of why we approach it the more traditional route with either the autogenous or other methods. But, you know, I'm sure that it's a promising route and certainly needs further research. But in terms of my specific project and our lab, as far as I'm aware, we don't have any plans for that as of yet.
And a somewhat related question, have you been able to identify genes related to the antibiotic resistance or virulence?
I personally have not. I haven't done any of the actual antibiotic resistance testing. This is all just built off of previous research, which I've gathered and then built my project off of. But yeah, I mean, I'm sure that there's something there, and it would be very interesting to see.
Awesome. Well, thank you very much, Sean. Great work and great presentation.
Absolutely. Thank you so much.